the spindle fibers. I repeat that since the plant cells lack the centriole, the Golgi bodies, remember the Golgi bodies are some of the structures or organelles that also form during interphase. They form more, and therefore they constitute or they lead to the production or the formation of the spindle apparatus or spindle fibers. Well, sometimes we call them microtubules, and that's not so important. So the centrioles pair and then they replicate. And as they pair, they start orienting themselves towards the opposite poles during interphase. Then the other activity is the energy production, and therefore ATP is synthesized. That's why we need more mitochondria for purposes of producing energy, which runs the entire process. Now here is the diagram of a cell undergoing the process of interphase, a preparatory stage. For example, in this case, you can be able to see the nucleus is fully intact and the nucleolus is visible, present, and almost to the center of the cell. And then the chromatin material appear as granules. The chromatin materials appear as granules. Then, of course, at this phase, this diagram doesn't have them. The centrioles begin to form. The centrioles begin to form, and then they start orienting themselves towards the opposite poles of the spindle apparatus. That is a cell at interphase 1. Now let's get to the real process of cell division, which begins with the prophase, then metaphase, and then anaphase, and then telophase. And remember, since we are discussing on the first meiotic division, those processes are denoted by the Roman numbers, either 1 or 2. But in this case, we are talking about the first cell, the first meiotic division. Therefore, prophase 1, metaphase 1, anaphase 1, and telophase 1. Now, what happens during prophase 1? During prophase 1, the chromosomes shorten and they condense. And remember, at this point, they are able to take a dye. They are able to take a dye. Then number 2 is the homologous chromosomes come together and then they lie side by side, a process that we call synapsis. Homologous chromosomes come together and then they form, or they lie side by side to form, or in a process we refer to as synapsis. Now, when they come into close contact, at some point the chromatids lie into intimate contact. Non-sister chromatids lying into intimate contact with a chromatid from a different pair, there is possibility of exchanging the genetic material. And that is a process that we call crossing over. So during prophase 1, there is the synapse, then crossing over, and remember, from crossing over, it leads to the variation. Crossing over leads to variation, as in the process there is the exchange of genetic material. For example, in this diagram here, we have this set of chromosomes. All of it, we call it homologous. Remember, from the word homo means same. It has similar lengths. These are similar lengths, and then they have the same form or shape, but the only difference is that they may have different gene locus. They may have different gene locus, and therefore the difference is only in genetic materials. Such a structure, we call it homologous chromosome. Now, you see in this second diagram here, we have two sets of homologous chromosomes. This one is different from the red one. Now, the two, they come into close contact, and this is what we call a synapse. a synapse. Now, since they are two different pairs, we call them bivalents. Bivalents. Now, at some point, or sometime, we've said that due to the chromatids coming into intimate contact, there is chance of exchanging genetic material leading to crossing over. Those points where they meet are the points that we refer to as the chiasma. For example, here at this segment here, the two chromatids. Remember, this is a different chromatid from a different homologous pair of chromosome with this one. So this is the point at which we refer to as chiasma for singular, and chiasmata for plural. Now, in this diagram here, you can be able to see the blue type of homologous chromosome has a gene locus for eye color, and at the same point on the corresponding homologous chromosome, at the same point, it has a gene for eye color. Remember, either one of them could be recessive or the other one could be dominant, but the two are housed at the same locality or at the same parts. So the points at which they come into close contact here is what we call the chiasmata. Now, therefore, crossing over is a process by which there is free exchange of genetic material between two chromatids of non-homologous pair. Homologous pair. And these can now be seen here. Remember, at the end of the day, now we have these two sets of chromosomes. They have some new strains. They have some new variation or some new traits. For example, this one entirely, as we started, it was blue. Then it has taken up some genetic material from the chromatid of a different pair. And therefore, this is what we say at the end of it, it has led to variation. So there are three very important activities that occurs during prophase. Number one, synapsis. Then number two, formation of chiasmata. Then number three, exchange of genetic material, and that will lead to variation. Now that marks the end of the first clip. Now we are going to discuss on the three others a short while.